Hello and Assalamualaikum everyone. In this video, our group will discuss about our research proposal entitled Coagulation Process for Pit Water Purification using Moringa oleifera. This proposal will be presented by me, Zetihan, matrix number 77256, and my group mates Manul Basria and Nur Atirakiana. This video presentation including introduction, literature review, methodology, expected outcomes, and work schedule. Without further ado, let's begin the discussion.
My name is Wan Nurul Basria binti Wan Muhammad Azhari. I will present introduction and literature review. This is the introduction for this study. Safe drinking water is vital to a community's health and well-being, and it must purify water from all sources before consumption. Due to this concern, coagulation process are favorable method of removing water contaminants due to the high efficacy and ease of use on various water types. However, this study faced problems that the use of chemical coagulants affect the treated water pH and produce large amount of non-biodegradable sludge and metal residue as stated by Nanfaji et al. 2020. There are 4 objectives for this study. The first one is to prepare the natural coagulant, Moringa oleifera, for pit water purification by coagulation process. Second one is to study the use of natural coagulants, Moringa oleifera, in a coagulation process for pit water purification. Next is to analyze the removal of some heavy metal such as lead, nickel, iron, and zinc from pit water using moringa seed. And last but not least is to evaluate the use of Moringa oleifera seed as natural coagulant for pit water purification. Next is literature review. This is the literature review for this study. The first point of view for literature study is a coagulation process for pit water purification. Coagulation is the process that combines small particles into bigger aggregates and adsorbing dissolved organic materials onto particulate aggregate so that contaminants may be eliminated in following liquid or solid separation procedure as stated by Kui et al. 2020. The second point of view for literature review is type of natural coagulant used in treating pit water. Some recent studies identify natural coagulant derived from mesquite beans, nirmali seed, and cactus latifaria cassia angustifolia seed, and many leguminous species as stated by Vijaya Yaragavan et al. 2011. The third point of view for this literature review is Moringa oleifera seed and background. Vilaseca et al. 2014 stated that Moringa oleifera lem, also known as the synonym Moringa ptericospermageton, is a tropical multifunctional tree that is originated in India, Pakistan, Asia Minor, Africa, and Arabia, and has since spread to the Central America, North and South America, and the Caribbean island. Almost every component of the plant, such as leaf, flower, seed, root, and bark, can be consumed or utilized medicinally or therapeutically. The seeds are eco-friendly, environmentally sustainable, inexpensive, corrosion-resistant to materials, produce tiny amounts of non-hazardous and biodegradable sludge, do not change the pH of the water under treatment, and are non-toxic technique of water treatment, making them favored over chemical coagulants, particularly in developing countries, as mentioned by NISA and Col 2020. Last but not least is properties of Moringa oleifera seed. Moringa oleifera seed have antibacterial properties, which make them effective against various microorganisms, as mentioned by Allah et al. 2012. However, several studies have shown that treating water with MO seed and extract reduce E. coli percentage, as stated by Sean et al. 2017 and Howe et al. 2018. The seed kernels contain a high concentration of water-soluble protein with a positive charge in a solution.
Next is the methodology. For the Moringa oleifera seeds preparation, we will wash the seeds that were collected and let it dry for a week. The oil extraction will be carried out by electrothermal soufflet using hexane. And we will use the jar test to measure the coagulation activities for the water sample. For coagulants preparation, the dry seeds of Moringa oleifera are then pulverized into powder using a grinder. Then we will use the Sofflet apparatus and hexane as a solvent to extract the oil content of dried Moringa oleifera seeds, and this will take around 8 hours. After extracting the oil, 100 ml of solvent which contains distilled water, 0.5 molar of sodium chloride and 0.5 molar of sodium hydroxide will be added to 5 gram of seed powder. Then we will use muslin fabric to filter the coagulant so that any insoluble materials can be removed before we conduct the jar test. Next is the water sample preparation. We will collect water sample from rivers near Kota Samarahan area, and we will test the water quality of the river using different sizes of Moringa oleifera seeds powder, and the performance of the seeds on river water will be evaluated by a jar test. For the jar test application, we will fill 8 beakers with 500 ml of water samples. Then we will fill 4 beakers with different grams of Moringa oleifera seeds. For the first 4 minutes, the stirring will be set at 200 rpm, then 400 rpm for 30 minutes. After that, we will let the mixture to settle for 60 minutes, and all the water parameters will be tested, and we will repeat the experiment with different grams of aluminium sulfate into another 4 beaker filled with water sample.
This is our expected outcome. For this project, it is expected that MO seeds or Moringa oleifera seeds will successfully prevent as natural coagulants. Besides, it is expected that the presence of MOC will purify pit water efficiently in coagulation process. The project also expected that Moringa oleifera seeds will remove heavy metals such as nickel, iron, lead, and zinc in the pit water. Last but not least, it is expected that Moringa oleifera will be reviewed or will be evaluated for other uses such as removing suspended particles, reducing microorganisms, and lowering turbidity in the pit water treatment. Lastly, our work schedule Gantt chart. This project will be started at August 2022 until August 2023. Our duration for this project is 1 year. At the first stage of our project, we will conduct the project research and finding, which we will collect the other studies that related to our project in 2 months. Then we will proceed to the second stages, which are fieldworks, analysis the water samples, experimental setup, and laboratory works. The objective of fieldworks is to collect the water samples. The water sample taken will be analyzed to measure the value of biological oxygen demands, total suspended solid, and chemical oxygen demand in water. Both of activity will be conducted between October until December 2022. Next is experiment set up in December 2022 until January 2023. And we will proceed to laboratory works where we will prepare the MO coagulants, water samples, and conduct the jar test. These activities will be conducted from December 2022 until March 2023. After finishing the laboratory works, we will proceed to data analysis in April until June 2023. Finally, the last one is the report writing and report presentation of our research project. This activity will be conducted from July until August 2023.
These are all the references that we use in this slide. That's all from our group. Thank you.